Hi there, and welcome back to another Microbiology for Critical Care Fellows. Today, I'm going to finish our sessions and discuss the gram-negative bacilli group. We've already talked previously about identification and the classification scheme and the general anatomy, and also more recently covered the gram-positive bacteria group. Today, I'm going to talk about the gram-negative bacteria, and since our emphasis is on medically important bacteria, I want to cover more depth on the Enterobacteriaceae group. Now, this group is important because it comprises the largest group of gram-negative bacilli that you're going to encounter in clinical practice. It includes a large number of medically important bacteria such as Serratia, Serratia, Citrobacter, Enterobacter, Klebsiella, Morganella, Proteus, Salmonella, Shigella, and Yersinia. This group is found throughout nature and is particularly ubiquitous in the soil and in water. In humans, it can colonize the gastrointestinal tract and particularly the oral pharynx. It only tends to do this, however, in patients who are either hospitalized or are otherwise immunosuppressed. Now, even though it will colonize these areas, under no circumstances should they ever be regarded as just part of the normal flora and should always be regarded as pathogenic. The exception to that is Escherichia, which when not producing nasty toxins, is resident in the bowels. Because it can cause infections anywhere, I'm not going to go into each individual organism and go in detail all the different types of infections it can cause, but just more generally cover those. But do understand that you can assume that you may find these organisms anywhere where there's an infection present. Particularly in patients who are known to be colonized with them because they are in hospital or because they're immunosuppressed. Because it's been a while since we talked about the anatomy of the bacteria, I just also want to go over this again, particularly focusing on gram-negative bacteria. The Enterobacteriaceae group, just like any other bacteria, has an interior that contains a single strand of DNA, and then also has a number of plasmids and ribosomes. The respiratory proteins are located on the inner membrane, and the inner membrane forms the insertion site for a large number of transmembrane proteins. Above the inner membrane is the periplasmic space, which is generally a very oxidizing gel, which helps it in defending against a variety of different noxic chemicals and proteins. And also within this space contains the peptidoglycan wall, which is, unlike in gram-positive bacteria, is a much thinner structure, but does help give shape and structure to the bacteria. The outer layer is important, and particularly in the Enterobacter group, because the outer layer is actually an asymmetrical layer of phospholipids similar to that which you see in the inner membrane, and also above that a lipopolysaccharide, or also called LPS. The reason this is important is because LPS contains a component called lipid A, also known as endotoxin. Endotoxin is a direct stimulant of the host immune system and acts primarily through toll-like receptor 4 on endothelium and immune cells to activate the immune system. Depending on the individual organism, there may be a capsule of varying size and composition, which is primarily there to help with adhesion and also to evade host defense. The pili, which are much more predominant on the gram-negative bacteria, particularly the Enterobacter group, are responsible for plasmid transmission, which is concerning, as this is how resistance genes are passed around. And also it also provides for cell adhesion to the host. So all of the Enterobacter group have a number of common virulence factors. They all have adhesion molecules on their outer surface. They're not necessarily for pathogenesis themselves, but particularly include things like papillae, which are not only just kind of sticky, but also have actual selective binding sites that allow it to bind to specific host receptors, which allow it to overcome the predominant negative charge that is present on most metabolically active cells. A number of bacteria within this group produce toxins. Most will produce some generic hemolysin, but some can actually produce a large number of enteric toxins. Now, you especially will see this with E. coli and Shigella, and this virulence factor can cause a wide variety of different gastrointestinal symptoms, from as minor to as a gastroenteritis to as concerning as a hemorrhagic enteritis with renal failure. Now, one thing that most cells and both prokaryotes and eukaryotes require is iron as part of many of the important energy and metabolic pathways. Now, part of the host defense is to actually sequester iron to keep it away from infecting invaders so that they can try and slow their growth. Unlike most other bacteria, the Enterobacter group actually secrete their own series of countermeasures in order to both scavenge iron and also overcome the host's attempts to scavenge iron to try to maximize its ability to acquire the iron that it needs to grow, reproduce, and produce energy. As I've mentioned before, one of the more important virulence factors is lipopolysaccharide, or LPS. This is the lipid A, or endotoxin, binds with toll-like receptor 4 as well as CD14, and is responsible for inducing the production of TNF-alpha, other cytokines, and also the generation of a large number of major histocompatibility complex receptors as part of activation of the immune system. They have a capsule layer that allows it to avoid phagocytosis, and then, as I've mentioned before, there's a large number of plasmids present within the individual organisms that allows it to pass on the bad news of resistance to not only organisms within its species, but to others outside its own species. Probably the most ubiquitous gram-negative that is out there in the world is Serratia coli. This is the one that can be a colonizer of your bowels and can be a quiet, non-troublesome creature, but can, when given the right circumstances, cause a whole host of problems. Probably the most common syndrome that it will cause is uncomplicated urinary tract infections, which are responsible for a large number of outpatient visits to family physicians. These are typically some of the bowel organisms from the host that have managed to make their way into the genital tract and then up into the bladder. One thing that is important to highlight about E. coli is the presence of resistance genes and presence on its plasmid that can induce the expression of extended beta-lactamase that confer resistance to a larger host of antibiotics. So as you can see here, on the left is the normal antibiogram for the majority of E. coli. in our area. And when you select out the ESBLs, you can see that the sensitivity to a large number of beta-lactamases drops dramatically, particularly ampicillin and ceftriaxone. But it remains still generally sensitive to carbapenems, but there are organisms in the Enterobacter group which have developed the ability to resist even carbapenems, which is obviously quite concerning given the ability of these organisms to pass on genetic genes to one another. When E. coli gets out of its normal environment and manages to evade host defenses, it can cause a whole host of other problems, including nosocomial pneumonias, cholecystitis, and also can be a source of peritonitis, and as well, once they're bacteremic, the possibility of seeding bones and joints is possible, and the development of osteomyelitis and septic arthritis has to be considered, particularly in the patients who have had some sort of a GI complication and then have developed E. coli bacteremia. Finally, there's an entire host of enterotoxins that E. coli can produce that can cause a wide variety of diarrheal syndromes that I'm not going to go into detail about today for the sake of time. Klebsiella is another of the gram-negative Enterobacter group, which has the ability to generate extended spectrum beta-lactamase inhibitors and become resistant to a wide variety of organisms. Again, as you can see, the changes in the antibiotic spectrum and sensitivities when an ESBL Klebsiella is present. Klebsiella itself, also like all the other Enterobacter group, can occur practically anywhere, including the urine, but also has a specific predilection for causing liver abscesses, and is not uncommonly found after there's been incidents of bowel compromise or instrumentation of the biliary tree can lead to abscesses forming in the liver. Proteus is kind of an interesting creature. It has the flagella that are more located at one of the poles, and is actually a fairly mobile organism under the microscope. So you can see whole hosts of them swarming around when you look at them in a wet mount prep, and kind of neat to see. Generally not typically a problematic organism, can cause a large number of different problems, as we've talked about above, but more to the point, it frequently will cause urinary tract infections, and this can be specifically problematic when there's a kidney stone that can then act as a source of a foreign body, allowing the Proteus to reside in a relatively sequestered location and cause ongoing infection. As with any patient who presents with a urinary tract infection, if they're known to have a history of kidney stones or have an active kidney stone present on investigation, you have to assume that the stone is infected, and you need to have urology involved to help deal with that stone. Enterobacter is a pretty nasty organism. It's primarily an opportunistic infection. It shows up in hospitalized patients, but has a large number of third-generation cephalosporin resistant factors, and can induce that resistance without a lot of pressure. So you don't even have to be giving them necessarily a third-generation cephalosporin, and you can start to see them in vivo start to express this resistance. It's a problematic organism because it also carries a large number of resistance genes, and that keeps it resistant ampicillin, and then also an increasingly resistant to ceftriaxone. This is the organism that has an inducible carbapenem resistance genes, and one that is probably a more important evolving organism in the world of antibiotic resistance. Now, Serratia is a much less common source of infection in the Enterobacteriaceae group, but I'd like to include it because it will show up from time to time. It can be a source of nosocomial pneumonias, but probably the coolest part about it is that it actually has the ability on culture medium, and as you can see on some bread, to form this red residue that looks like blood. Serratia is relatively sensitive to a wide variety of third generation and carbapenems, and resistance is not generally considered a huge problem as of yet. But as I've said before, it's an Enterobacter, part of the Enterobacter group. It shares many of the common characteristics with all the other members of the group, and can certainly become an evolving problem given time and antibiotic pressure. So the last gram-negative bacteria I want to cover today is Pseudomonas. Now, you will see on your initial culture reports, gram stain will say gram-negative bacilli, and then a follow-up will say, lactose fermenting or non-lactose fermenting gram-negative bacilli. If it's a lactose fermenting, then it's part of that Enterobacter group that we've just been talking about. But if it's a non-lactose fermenter, then you need to think that this is probably Pseudomonas. The entire Pseudomonas genus is another ubiquitous organism in both throughout the environment. It's present anywhere, typically inhabits soil, water. It's in plants, it's in insects, all kinds of animals. It's everywhere. The only thing of interest of that, though, is that it actually is not a colonizer. And in a healthy human being, it does not colonize a healthy human being. So if you find it on somebody, that's already a problem. Much like the other gram-negatives, it has a number of virulence factors that allow it to remain resident and also to evade host defenses. It has a large number of pili, which, as with other pili, allow for it to adhere to surfaces, and it also can produce its own biofilm that allows it to duck and cover from the phagocytosis. It also has a series of secretion systems that allow it to eject toxins into the extracellular space. And also, interestingly, it also has a protein that allows it to actually inject toxins directly into host cytoplasm and actually cause cellular damage by essentially a hypodermic needle attack. It also produces a number of quorum sensing molecules, which are really fascinating. These are molecules that are produced by bacteria that form a signal that signal to other molecules around, or other bacteria around it, that allows for communication and coordination of the bacteria. You can get a large number of bacteria that will sort of stay resident within an area until it senses that they have a sufficient, essentially a sufficient number to engage in active infection, and then will secrete enough, will communicate with each other. Yes, we have enough. Let's go, boys. In we go. And it's these quorum sensing molecules that are responsible for that. There's quite an excellent TED talk available by Bonnie Basler on how bacteria talk that goes into a lot of detail about this, and is really quite fascinating, especially since it has is providing us new sources of treatment against against invasion. Another excellent talk that Bonnie Basler has produced is also called The Secret Social Life of Bacteria. And I would recommend both of those to you very highly, as they're quite interesting and very excellently done. Pseudomonas has a number of ways in which it can resist antibiotics, both intrinsically in the way it's built, and also acquired methods. It has intrinsic resistance to a large number of antibiotics, as you can see here, and a lot of this is due to the decrease in permeability of the outer membrane, the presence of efflux pumps, which will actually just eject anything that's directly toxic, and then enzymes within the cytoplasm that allow it to, when exposed to antimicrobials, to actually modify the molecules and make them harmless so they don't have their effect. As with the other gram-negative bacilli, it has the ability to accumulate plasmids and therefore has the ability to accumulate genes resistant to a wide variety of different antibiotics, as well as ones that develop an intrinsic resistance. There are also genes that allow aminoglycoside modification and encode for enzymes that will essentially neutralize aminoglycosides, which is critical as aminoglycosides are one of the mainstays of dual antibiotic therapy for Pseudomonas. Pseudomonas is ubiquitous in the environment, and it's not really so much of a problem until you get the person into the hospital, and then it can become a very serious issue because of its polymicrobial resistance. It can cause any number of different problems, including all the other sources of infection, causes of infections that we've listed above. It can be present anywhere. It can also cause skin and soft tissue infections, which is a little unusual for the gram-negative group. Finally, you can occasionally get outpatient infections with Pseudomonas. In the case of swimmer's ear, that's or otitis externa, that's a pseudomonal infection, generally reasonably self-limited and actually usually fairly sensitive to treatment. Probably the most common and most famous infection Pseudomonas can cause is hospital-acquired pneumonia. Like other enterobacters, it has a large number of resistance factors, and therefore this specific organism you need to consider dual coverage to help effectively treat it instead of picking a single antibiotic. And that's all there is to it for gram-negative bacilli. As we've discussed, there are two main groups, the Enterobacterales group and Pseudomonas. This is distinguished by their use of lactose, whereas Pseudomonas is a lactose non-fermenter, doesn't use lactose. Enterobacter group will use lactose as a lactose fermenter, and this will be your clue that you'll see on your report. There's a wide variety of species within the Enterobacter group that cause a wide variety of different infections, and unfortunately, based on their intrinsic structure and their exposure in the environment, are developing an increasing level of resistance to all of our therapies. So on a final note, I want to again thank you for listening, but remind you that when you're dealing with bacteria, it's important that you know your local antibiotic sensitivity patterns, and your microbiology lab should be producing an antibiogram that should be available readily to allow you to help effectively select the proper antibiotic sooner, so that you can then start tailoring therapy to antibiotics that do not induce other resistance. As always, if you have any questions or comments, feel free to contact me, or you can leave comments in the section below.